Thank you so much. Well, thank you all for coming. It is a great pleasure and honor for me to come here and to really witness and participate in the Appalachian Student Forum. It's been wonderful so far. What a remarkable celebration of creativity, innovation, and science, and excitement, which really all of you who contributed to writing and displaying these wonderful posters, just thank you so much. I've been going to a relatively large number of meetings recently, but never I've been to a meeting which is as diverse, both from the participants' point of view and from the content point of view. This morning I looked at the posters which dealt with issues of spirituality and issues of pancreatic cancer, very local issues like lack of primary care physicians in Appalachia, to the global warming issues, from psychology and behavioral sciences like fear of personal rejection, to really an intricate mysteries of plant biochemistry, plant biology. Congratulations to you all. I learned certainly much more coming here than I would have learned going to a regular scientific meeting where everybody knows each other and we all know about the subject we're going to talk about. Thank you so much. So this afternoon I'll give you a talk which is going to be a very broad one. I will tell you a little bit about what we're doing in the lab, but mainly I will talk about the magic of plants and human health and the connection of plants and human health. And tomorrow morning I will be talking much more specifically, much more scientifically about specific botanical products for human health we are studying in our laboratory at Rutgers University. So I'm going to be talking about these botanical therapeutics, which have been with us from the dawn of civilization. Basically, plants which bring more than food to us, which bring health to us, and bioactive compounds from plants which help us prevent and treat the diseases. And remarkably, we have fossils, we have frozen human remains from both Andes in South America and from actually Alps in Europe, where 4,000 years old, those people were frozen accidentally there. And actually, in the case of a man which has been recently uncovered in the Alps, he had with him this pouch which actually contained extracts of plants. Actually, it was fungal extract, which apparently he carried with him to help with some maladies. Actually, we know now that he was infested with worms, and that is, we know that what was in this pouch actually had some antihelmintic activity. It's the extract of the fungus from the birch tree. Every civilization which existed on Earth and left a written record actually have left a record of plants and human health, from Babylonian civilization, from Ur to India, or Vedic medical system, Egyptian, Middle Eastern, of course Chinese. Human plants and health will be always connected. A lot of, through the Dark Ages, I think a lot of knowledge has been preserved in the Orient, in Arab countries. Then, of course, during the Renaissance, it moved back to Europe. 18th century, we're truly starting to use scientific methods in our study of botanical therapeutics. And I'll give you some examples, which I think the most remarkable, particularly which deal with the first, as far as I'm concerned, true discovery of a functional food by James Lind in 95, actually, to the withering and the digitalis extract. 19th century, we started to do really good chemistry. We were able, actually, for the first time, to isolate the bioactives, whether it was salicylic acid, quinine, morphine, and many other compounds from plants, identify their structures. And beginning of the 20th century is truly the birth of modern pharmaceutical industry as we know it. But this connection has been in existence for a very long time, and truly this is the first time we're living in this couple of generations when we don't see plants as something immediately valuable for health, because of course pharmaceutical industry, and this is not meant as a bad thing at all, but clearly they have separated the natural products from the synthetic products, and now we can synthesize a lot of molecules forgetting where they originally come from. So this is one of, to me again, because we are very much now interested in functional foods and health, and I will tell you a little bit about it. This is to me a very important person, actually a Scottish physician by the name of James Lind, who've done two most remarkable things, I think, to medical sciences and to functional food area. He was the first person ever who really established the value of clinical trials. And secondly, he discovered the first functional foods which truly was able to save thousands, if not millions, of lives, because scurvy, which is a deficiency, vitamin C deficiency, which softens your cartilages, breaks your bones, creates bleeding, and responsible for scurvy, was really a disease which was killing, was preventing the travel, long-term sea-going travel. James Lind, for the first time, thought that maybe it has something to do with the sailor's diet. He truly performed the first, to my knowledge, clinical trial, which was well-deserved, and really changed the way medicine is. He basically selected 6 groups of sailors, and he gave them different treatments each day. Cider, garlic, mustard, seawater, sulfuric acid, vinegar, and then one group, group number 6, received 2 oranges and a lemon. And before that, we didn't understand anything about these clinical trials. And what he's seen was quite remarkable. I mean, they ran out of lemons, but actually they didn't last 12 days, 14 days. On day 6, they've seen such a remarkable effect of lime juice that they already figured out, what is going to be the treatment for scurvy. Unfortunately, that was 1747, and 1795 only was the year when actually British Navy started to give sailors green vegetables and citrus fruits to prevent scurvy. That was because subsequently he made a mistake. He decided to actually make a more active product from lime juice, and he started distilling it and boiling it and evaporating it, creating an extract. He didn't realize that in the process, actually, the vitamin C, the ascorbic acid, has been destroyed. So actually, that was a problem. But still, it's a remarkable discovery. It's the first clinical trial, and now we know. I mean, we do exactly the same now. Without clinical trials now, we cannot really establish the efficacy of any pharmaceutical agent. 1785, another British physician, in this case it was Bill Withering. For the first time, I think it's not that he was the first one who discovered the botanical. They existed before, but really used the scientific approach to identify the botanical and to validate it, and I think to create a standardized version of it. He was exposed to this knowledge that a collection of herbal extracts containing digitalis, the foxglove, actually is good for treating some cardiovascular diseases. Dropsy, which is basically edema associated with the retention of water because of poor cardiovascular activity. He was able to, as we would call it now, dereplicate, deconvolute this mixture, figure out that digitalis is actually the plant responsible for it. And for 9 years studied the best ways to actually prepare the standardized form of digitalis, which turned out to be a green powder of young leaves, which truly also and it is almost as active. That form he developed is almost as active as a digitoxin, which actually still is a part of some of the drugs which being prescribed to the people. And then this is what really gave birth to pharmaceutical industry. And that was a plant. And of course, it's some dear plant to some of us because it's a plant which contains salicylic acid. That's a willow tree. And willows have been used, actually, the bark from the willow tree. It doesn't contain salicylic acid as much. It contains salicin, which is an alcohol of salicylic acid. It's shown there. But it's been actually independently discovered in a new world and an old world that the bark of willow tree is good for aches and fevers. And actually, Lewis and Clark, based on the traditional Native American knowledge, carried willow tree bark and willow tree bark extract with them on the expedition to the west to treat their problems. And the problem with salicylic acid itself, as was discovered by Bayer, which is a pharmaceutical company, was that actually it's too toxic. It creates this intestinal ulceration. So in 1895, actually, they've developed a synthetic form. They acetylated salicylic acid synthetically. Aspirin is not a natural compound, but they took a plant product, created an acetyl form using this reaction there, showed that this is also as effective, but it has less side effects. In 1895, it was for the first time, the first actually synthetic single molecule pharmaceutical was placed on the market. It's still the historically the best-selling drug ever, by far, because it's been around for so many years, and still you can buy it in almost every pharmacy. So this is just a short contribution of plants historically. Again, if you look at what's available now, 25% of all pharmaceuticals which are on the market now directly or indirectly derive from plants. Alkaloids, and for those of you who already had plant biochemistry, I'm not going to ask what alkaloids are. Those are nitrogen-containing basically compounds from plants. They're the most diverse group of plant molecules which made it in modern pharmaceuticals. More recently, terpenes and steroids, digitalis we already talked about, and cardiac glucoside from it, but compounds like Taxol and artemisinin, which is actually sesquiterpene lectone for malaria, has been developed, and then the phenolics and polyphenols. And actually, those are not compounds which are commonly used in drugs, but this is, I think, an extremely important group of compounds, very diverse group of compounds, which are very important for dietary supplements and functional foods, which I'll be really talking to you. When you deal with medicine and human health, regulations are very important. That's a big mess worldwide because each country regulates things differently. But this is really the categories. These are the legal categories in which we put botanical therapeutics in this country, in the US. Those are mainly developed by FDA and they're managed by FDA. The compliance is managed by FDA most of the time. And if you are doing research, which is discovering, validating, developing any of the botanical therapeutics, I think it's best to realize which category your product will fit in, because that truly determines the path you're going to take to prove its efficacy and hopefully have it delivered to patients and people in need. Of course, we talked about drugs or ethical drugs, common pharmaceuticals. They call NCEs new chemical entity. This is just a jargon we all use because it's one single molecule, which is usually new, has to be new so it can be patented. And that's, of course, a problem sometimes, because non-patented molecules are very difficult to make drugs from. But those are the things we are familiar with. Those are the ones which have been on the previous slide. And then we have something which is quite dear to my heart, and those are multi-component botanicals. Those are botanical therapeutics which are not really a single molecule, but rather they're always mixtures. Botanical drugs is a new category which recently FDA has approved, 2004. Only 2 products on the market. Verogen, which is an extract. Those are real drugs. They've been developed for IND and DA process for clinical trials. They've been approved as drugs, but they're plant extracts. Only possible from 2004. Verogen is an extract of extract of tea for genital warts, and crofelemer has just been approved in January of this year. Actually comes from a South American tree called the blood of the dragon. Some of you may have known it. It has this dark red juice. And actually this is for the treatment of diarrhea associated with HIV. That's been approved as a drug, and that's a plant extract. And then there are dietary supplements, which are, of course, nutraceuticals. We all know them. Food additives, medical foods, again, rather complicated category which exists. Functional foods, cranberry juice, your oatmeal, those kind of things, which actually have health-related claims. They're not just there to promote health, to give you calories. And then cosmetics. You've seen probably lots and lots of botanical extracts in cosmetics you buy, and of course, they usually put there by sort of us unscrupulous marketers, rather because they do have some really proven clinical efficacy. Some of that is true for this loosely regulated category of dietary supplements and nutraceuticals, all those botanical extracts, which still are, their sales is mostly are based on marketing rather than really hardcore knowledge. But I think this is changing quite rapidly. The reason we're so fascinated with plants, because they're just absolutely remarkable chemists. If you look at a plant genome, there's lots of genes there, just as many as we have, if not more, a human being has. And lots of them encode very intricate biosynthetic pathways which actually produce those natural products, those bioactive compounds. Because plants cannot run away, they cannot fight, they don't develop this fight or flight behavior. They sit there exposed to all elements, and they behave as chemical chameleons. They're constantly respond to the environment in the only one way they know is by selectively inducing the synthesis of certain compounds, hoping that they'll provide defense against pathogens, against herbivores, things which try to eat them, against high UV, against light, against low light. It is all done through chemistry, and they've learned how to do it extremely well. And actually, I used to work for DuPont, which is one of the largest chemical companies in the world. I worked with plants there. And you come to realization the single plant you step on every day when you go to work actually makes more chemicals inside it than probably all chemical industry in the world. I mean, if you count nucleic acid, peptides, protein, all the natural products, you're probably talking about 80,000 to 100,000 compounds in each plant being there all at any given time. And this is quite a remarkable chemical ability. And those compounds are also there because they have a function, or we assume they have a function. They're optimized to actually interact with certain biological targets. And that's why I think they make particularly good pharmaceutical agents sometimes. There's been a lot of controversy how much is really left there in our meadows, under the canopies of the jungles, in the oceans. How many undiscovered drugs are left there? And I know I don't have a right answer for it. I do not know, because very few of those really plants have truly been explored. Certainly, if some of them have been explored for 1 or 2 clinical endpoints, but certainly not for a tremendous amount of targets which we now have in our pharmaceutical discovery. There's been a lot of work which has been published in the 90s and 80s suggesting that about great majority, 80 to 90% of phytochemicals which can make drugs are still out there. They're still hidden from us. And we keep discovering new ones, and we keep approving new ones because, for example, artemisinin, which comes from traditional Chinese medicine used to treat malaria. At that time, didn't know the name malaria, but that comes from Artemisia alba, traditionally been used for centuries, but it's been actually approved by FDA only 3 years ago in the US as a frontline defense against malaria in combination with other anti-malarial drugs. So those things keep coming. But the pharmaceutical industry, again, I'm not blaming them. I think they have a reason to. Clearly, they're losing interest. They're losing interest. Everybody's losing interest in using natural products, not just plant natural products, but natural products to discover new pharmaceuticals. And the reason is because this collecting things from the environment, from nature, from plants, from microbes, from fungi, has competition now. We've been very effective. We've developed chemical synthesis over 100 years ago. Combinatorial chemistry and related technology which allow us sort of create a big libraries of synthetic compounds in a very short period of time. That's a technology which competes directly with natural product sources. Computational type approaches, the docking type algorithms allow us actually to design a molecule which will dock to a special receptor site. And then there's all these political issues which makes it very difficult for people to go and collect plants in different countries. I'll talk to you about that, and our solution to that basically led to a major decrease in interest in natural products as sources for new pharmaceutical discovery. And of course, all of us who have vested interest in plants said, well, look, but really, if you look at the number of successes of those competing technologies, they've been not great at all because the value of pharmaceutical companies are going down, the stock values and the number of new drugs on the market is actually dropping every year. So, and this is truly a survey which I think is extremely telling, been done by 2 really great, colleagues, and really, colleagues is probably not the right word, but role models for me in this whole thing is Dave Newman and Gordon Craig from NCI, National Cancer Institute. Spent their careers developing natural product-based drug discovery products for programs for NCI, particularly Gordon. And they've been tracking the sources of all pharmaceuticals which are being commercialized every year by all pharmaceutical companies, whether they come from nature or whether they come from purely chemical, sort of non-rational type design, or their natural product type. And they've been publishing those papers. Recent one came out actually in 2012. And I'm not going to go through this. You can read it. But this is really the conclusion from this paper. If you look, this is what I already told you. The total number of actually small molecule-based drugs has been going down every year. all these competing technologies were not that spectacularly successful. Of course, biologics are coming up, vaccines and biologics like proteins, but still. But here is an interesting thing. The funding for natural product-based drug discovery has been drying up. And yet, the number of compounds which are derived, and this is actually the summary of all compounds which are derived from nature, just compounds which have been isolated directly from plants and other natural sources, or been isolated and modified, or the pharmacophore comes from natural product. Those numbers, have been absolutely constant for all the years they monitored this. So there is still absolute value in natural products as sources for drug discovery. It probably will never go back to just going, collecting plants and screening them, because the modern discovery technology, high-throughput screening, is just not compatible with small number of extracts you can produce. They have insatiable appetites for samples, and this is just something you cannot produce from nature. But nevertheless, up to today, the new pharmaceuticals based on natural product designs remain pretty constant. I'm going to skip that and sort of talk to you a little bit about how we are addressing some of those challenges, because I think there's still an opportunity to connect plants with human health. It may not necessarily be through the discovery of novel skeletons from nature and from plants, which will be new drugs. I think those skeletons are very important, but it's likely now that pharmaceutical companies will take them and start decorating them, improving their bioavailability, efficacy, which is, I think, a right approach. I think there are very few molecules which will be intact, which come from nature to become the new pharmaceutical agents, but they'll be always an important source for the new discovery. But here is food, and if you look at the agriculture and agricultural evolution, I think also what we grow, how we grow it, how we select it, and what we buy have changed, certainly in this country, certainly in the developed world. Because we started with just subsistence. I mean, we just grow, and this is all just really deals with how much we can harvest or how much we can grow, and trying to basically fill our stomachs. Well, somewhere 2,000 or 3,000 years ago, people actually started to do actual breeding for yield, basically for calories. They try to increase the size of the grains. They try to increase the amount of protein. They didn't really know what they're doing, but they succeeded in increasing oil content. They actually, at the same time, they succeeded by breeding many natural products out of our foods because they usually don't taste well or they have some deleterious effects. So we were just, this whole 2,000 years of yield breeding basically resulted in breeding for oils, starch, and proteins, with all those bioactive molecules being bred out of our crops because we didn't want them there. Essential nutrition, really for things like vitamins, for things like specific oils, for things like better quality proteins. Well, maybe that's about 100 years ago. I mean, of course, things like golden rice, you all know that's the molecular approaches we used. But of course, very recently with the whole food and all this, we were really truly studying. I think we went back to what Hippocrates said long time ago. And now we're, for the first time, truly starting to breed, engineer, and buy plants to really be our medicine. And for the first time, starting to really produce scientific methods to validate that. So I'm going to talk to you about one group and sort of getting more specific in what and how we do things. Polyphenols, because berries, fruit, vegetables, these are your big sources of polyphenols. They're known by general public as antioxidant. This is a term which has little scientific meaning. I mean, it has a chemical meaning because we believe, and many of my colleagues believe, that polyphenols not just act because they're antioxidants. That may be a part of the reason why they're so good for you. But they also certainly have specific modes of actions and specific targets which have been identified for cardiovascular diseases like green tea or chocolate compound, anti-inflammatory like quercetin, and just many others. But general public thinks about them as antioxidants. There's a very large group, probably 4,000 is underestimate, probably there's about 8,000 of them around. And they have all kinds of activities which have been scientifically documented. I will specifically talk about anti-diabetic and anti-obesity because this is something which our team is particularly interested in. And we have actually programs, NIH-funded programs, which can take compounds and botanical therapeutics with them all the way to clinical trials. But they've been also, of course, reported as anti-inflammatory, anti-tumor, and just from vision improvement to cognitive improvements for blueberries and so on. Here's the issue. As I told you, we bred a lot of those things out. So how do we concentrate them back in? How do we actually, in one serving of a food, if we want to really make truly functional food, which is good for you, for example, very high polyphenol food, which is even more polyphenols than blueberries, for example, which has been sort of our gold standard so far. How we do that, how we put it all back. And this is a major challenge of this, because the way we know how to do it is this. We all do it very well. Well, we can just take an extract of a plant and we can isolate those polyphenols. But, as I showed you before from that regulatory FDA table, you're stuck with that now. It is no longer food. It is no longer dietary supplement. Now you cannot add it back. And the reasons for it are very good. If you concentrate compounds so much, they're pure compounds, they're solvent residue, all this, I mean, they may be toxic. So we're stuck with writing scientific papers about all those extracts, which many of us have isolated and published, thousands of papers on this. But this is not a way how you can actually take advantage of concentrated molecules. So we've been working on something very, very simple and very trivial, which is maybe almost unimpressive, but it works. We've been trying to find a natural way in which we can concentrate polyphenols in foods, which will still keep it as food. So usually we were limited to this kitchen sink chemistry. We couldn't do anymore. We couldn't use solvent. How to concentrate polyphenols from, for example, blueberries, which we know are good for you, or cranberries, or grape, while keeping that final resulting product still as food. Why did we want to do it? Well, the reason is simple. Blueberry, for example, polyphenols, we know they're anti-diabetic. If you isolate them and you give them to mice, dogs, whatever, rats, or people in clinical trials, they will increase the insulin sensitivity and there's certain anti-diabetic benefits to them. And there's a lot of antioxidant power in them. But again, you cannot do that as food. The problem also, if you just eat blueberries and you have diabetes, well, you get those anti-diabetic polyphenols, but at the same time you get so much sugar that it almost negates the effect of those compounds, because all berries, polyphenols in fruits that just coexist with sugars, which have pro-diabetic, they're hyperglycemic. So how do we concentrate them? And this is a very simple solution. I'll tell just a little bit about it. We basically thought about it and said, well, can the plant proteins like soy protein, which is one of the most cheapest commodity we have, which is insoluble, tightly packed, basically mini particle, which has a lot of surface charge, can that serve as actually the affinity resin on which can bind and concentrate polyphenol? So we make this affinity resin, which is food, and then we try to bind it with polyphenols. We also remembered something which has been invented by human civilizations thousands of years ago, called leather tanning. What is leather tanning? It's when you take a bark rich in polyphenols from oak tree or any other tree, you make an extract and you bathe leather in it. What happens, polyphenols will bind to proteins. We know that much. And making leather supple. That's the whole idea. So anyhow, this is how it looks. We can take a soy protein isolate, which is a pretty much standard commodity. You buy it in tons. You can buy it in railroad trucks. And you can do it at home. And this is a blueberry juice concentrate, which you can also buy. It has lots of sugar in it, extremely sweet, but it also has those anti-diabetic polyphenols. You mix it together on the right pH and temperature, and what happens is now indeed the polyphenols will bind to the soy protein, leaving this spent juice behind. You can concentrate it, make all kinds of stuff with it, but it has lots of sugar. But sugar does not bind to protein. It's just way too polar. Only mid-range polarity compounds will bind. So you get this, what we call it a Nutrisorb process. You get this really, and I use blueberries because you can see color, the matrix, which really has high levels of polyphenols. It is so high to basically just give you an equivalent, a serving equivalent of 1 cup of blueberries. We'll take 4 grams of this matrix. For grape, it's only 2 grams of this soy protein, grape polyphenol, naturally produced, basically still food matrix to deliver equivalent of 1 serving of grapes. Well, for cranberry, we don't eat cranberry. We drink cranberry juice. So That's a big serving size, 250 milliliters, so that will take about 5 grams if you compare to juice cocktail, which most of the women will drink if you have UTI. Oops. And again, I don't have time to show you all the data. I'll just show you very little. I don't want to put lots of data in this. We've done a lot of preclinical and clinical work with the efficacy of this, what we call sort of this Neutralsorption process. We used diabetic mice models, acute and chronic. We have that all in the lab thanks to the NIH support and thanks to the fact that I'm a member of NIH Center for Botanicals and Metabolic Syndrome. And you can readily show that if you take a rat or a mouse which has been really fattened with a high-fat diet, sort of like McDonald's Western-style diet, and it becomes diabetic, we have those systems. And you give it the blueberry Nutrisorb or just soy protein as a control. If actually it's been Nutrisorb, you see that the sugar level in blood will drop very rapidly, almost as well as the metformin level. Metformin is a drug. This is what doctor will probably prescribe to you if you have type 2 diabetes in the early stages. So quite effective, probably very effective because there's no sugar in it. We've done long-term feeding studies showing the same. Actually, animals will also reduce the weight. They don't become as obese, and they are also at the same time less diabetic. So this is a very interesting kind of observation. And we were wondering why is it so effective, actually, if it's complexed with soy? And we are fortunate we have this human digestive system model in our lab. It's called TIM. It's sort of all the tubes. They mimic different compartments in our digestive system. They have all the juices, all the enzymes. They have peristaltic movement. I mean, it's just the company who developed it. They work with Rutgers. And we were, to our actually amazement, we saw that polyphenols, because they're protected by the protein, this binding protects them, they actually are more bioavailable. They are protected from saliva, and then they become, you will see them much more of them as being bioavailable. So actually more of them, and we've done animal studies, more of them go into the blood. And it also increases their stability. So you can, because polyphenols are, so there's a lot of benefits which this complexation gives you. So we just hope that in the future, those kind of ingredients, and we are starting to work with some interested companies, food companies, which actually develop the foods, functional foods out of the blueberry, grape, cranberry type Nutrisorb. But we always are interested, and I will spend probably the rest of my talk more on this international component. We're also interested in international work. And so immediately when something comes up, we see, can it be applied to the developing world, to the developing countries? And we realized then that one thing Nutrisorb can be useful for the developing world, and actually the ability to fortify the basic staples, because there's a lot of flours, a lot of legume flours, soy flour, pea flour, chickpea flour, all those high protein flours, which basically give you protein but nothing else. At the same time, in tropical regions, you have a lot of fruits which are being produced and vegetables. But the problem there that is a tremendous seasonal post-harvest losses associated with those. For example, mangoes is something which we know quite a bit about. Almost 80% of it is lost because there are lots of mango trees. It's a very rich source of provitamin A and polyphenols, but they will ripen around for 2-month period, and then they just rot in the ground. Few of them get captured. So we approached Bill and Melinda Gates Foundation and said, well, can we use Nutrisorb technology? And because of our connections in Zambia, we wanted to use Zambia. Can we actually use that to help to create this fortified basic foods where the fruits which will go to waste, we can just juice them, bind the bioactives in the flour, and then that can be used in traditionally acceptable porridges and other products. And we were fortunate actually enough to, in collaboration with Dr. Marianne Lila in Kannapolis, she's the director of Plants for Human Health Institute there at North Carolina State University. We've developed actually the process, this is just mango, how very simply you can, women in the villages can connect mango fruits, juice them, add flour to it, just throw away and then the spent juice, and then use this flour really enriched in vitamin A and this carotenoid-like molecules which are in mangoes, but also with other polyphenols, just in the porridges to really improve the health and immunity of particularly women and children. So this was a very nice application, which we hope will take root in the developing world. Here's the other approach we're using for creating foods which have higher levels of polyphenols, beneficial polyphenols. This is different. This deals with what we call nutritional breeding. It's non-transgenic because I think transgenic approaches to that are going to fail because of the regulatory environment. It's just not, I mean, for major crops, yes. I'm not a basher of genetic engineering at all, actually. I've been right there when it was born. But it's just not happening. I mean, transgenic vegetables and fruits are not going to be with us in the near future. Just the outcry is just too much, particularly in Europe and certainly in Africa and in India. So we decided, well, can we just look at lettuce? We just love lettuce. Lettuce is nothing good about it, there's nothing bad about it, and everybody eats it. And it's an enormous market, and it's just sort of there. Don't ask me why. We just eat it culturally, and this is, after potatoes, probably the second most commonly consumed vegetable in the world. It's not known for anything, but it's a plant. It can make polyphenols. Actually, there are red lettuces out there which has quite high levels of polyphenols. So we were fortunate enough to attract a really great tissue culture biologist to our team, Natasha Pogrebnyak, and she'd been using summer clonal variation combined with some mutagenesis approaches and a color as a marker for polyphenols because of the anthocyanins, as are polyphenols, to really look at many, many clonal variants in tissue culture of standard lettuces to see whether we can come up with which is much redder than others and also have other polyphenols in it. And it's been 2 years worth of research, and this is Natasha with what she produced. We call it Rutgers scarlet lettuce. Rutgers actually allowed us to use that name. It is almost black. It tastes perfectly fine, but this is really what I think is so remarkable about it. And we had it tested by independent labs just to make sure that we are correct. This is the level of phenolics, milligrams per 100 grams of fresh weight. Those are just anthocyanins, which is the color. And this is blueberry. This is our golden standard for phenolics and for ORAC, which is a measure of antioxidant activity. Okay? And if you just take, and remember, that has lots of sugar in it associated with its polyphenols and the antioxidants, which is really the same. This lettuce has no sugars, but the antioxidant capacity of this lettuce per weight, per fresh weight, is over 3 times that of blueberries. And also polyphenols about 2.5 times higher. So now, we have something which may actually be a functional food and give you those compounds in levels which are sufficient to do something good for your health. Of course, all of that needs to be validated for clinical trials. I'll go back to James Lund and just have to do it. But this is an interesting beginning, and there's a lot of interest in those. And again, we showed that this is anti-diabetic in all animal models. Here is an interesting part. I mean, if you really try to, and this is a biochemical profile, which we've done on that. And one of the most desirable compounds actually for diabetes, which has probably most validation behind, is chlorogenic acid, which actually decreases the release of glucose from the liver. That's well known. Actually, that's one of the reasons there was a recent clinical study which coffee may be good for you, and actually been shown that the consumption of several cups of coffee a day will reduce your chances of becoming diabetic. But we have, somewhere underneath 3% of chlorogenic acid in this lettuce, which is very high. It's almost the compound we would like to have. We have it, and it has quercetin, also very high amounts, and it has anthocyanins in it. So those are the stuff. So those are good molecules to have. We know what they are. They're good polyphenols. This is actually the cultivation of Rutgers scarlet lettuce by farmers in New Jersey. And so the last part of the seminar, I want to switch to what we do, to how all of us can help the world. Again, I don't think we can compete with pharmaceutical companies in developing new drugs. I think it's not something small university lab can do. But if we really want to help to discover and develop novel botanicals, to really utilize this putatively undiscovered value of those wonderful compounds which are still hiding out there, what can we do? And we formed about 8 years ago, we formed with Rutgers and University of Illinois and now North Carolina State University. We formed the organization which is purely philanthropic, which is called Global Institute for Bioexploration. And the reason we did it is because before 2004, we were involved in this conventional bioprospecting, and this is something with NIH like to fund. And actually, we were funded quite significant amount of money to work in Central Asia, Uzbekistan, Kyrgyzstan, Tajikistan, and Uzbekistan, to basically collect plants, extract them, form partnerships with pharmaceutical companies here, take the extract out, sign all the documents of equitable benefit sharing. We had the Rio Convention, which truly, attributes, that gives the rights to all biodiversity, all the plants, to the country. So country has now, by law, sovereign right to their biodiversity. U.S. never signed, actually, the Rio Convention, the Biodiversity Convention. I think Vatican or Iraq also, 2 other countries. But we comply. We do comply with the law, and certainly NIH does. So, but we quickly realized, Central Asia is very totalitarian places. You can do a lot of things there, but we quickly realized that it's just not a paradigm which can work in the world, because there's this tremendous resistance to what is perceived to be biopiracy when us going there collecting plants, stealing natural resources. And the Rio Convention, it was not intended by it, but that was an unfortunate consequence of it. It actually made it more difficult because to do those kind of things, because people started to realize that maybe truly something is in there, although I do not know of any new drug which has been actually isolated from the plant in the last 20 years, for example, you're doing bioprospecting. But anyhow, this is a reality, this is a perception, and we just can no longer do that. So what can we do if we are still interested in biodiversity as a source of new pharmaceutical agents? Well, this is what we can do. We can totally reverse the flow. We can reverse the paradigm of how do we discover things. So now, rather than taking the materials out of the country, possibly we thought, with my dear colleagues, we can develop some simple screens, some simple enabling technologies, simple assays, which we can actually take to the countries, train scientists, students, villagers that will be so simple. So they can at least do this very early step. I know this is not a way to develop drug, but at least to validate the traditional knowledge, to catalog the biodiversity from the functional point of view, to at least start that discovery process, and at least take it to the point where they can own the intellectual property. So then it's easy, because the big issue in this whole bioprospecting, who owns what? And this is what the argument is about. If there's a clear ownership by, for example, developing countries, even companies like Merck and Bristol-Myers, fine, they'll license it. Then it's easy. Then there's no controversy. The controversy is who owns what because of the traditional knowledge. So take it to the point where a country can have the intellectual property, and developing the simple assays. We call them screens to nature, because before that the paradigm was nature goes to the screens, to the assays. Can we develop the simple screens which we can take to the developing world? And there's been some wonderful scientists, creative people who work with us, who created this manual, which sort of Rutgers claims ownership to, which has about 17 different very, very simple assays, which all of them can be operated from the pickup truck using the basically cigarette lighter, and that was a requirement. I'll just show you some of them so you understand what they are. Actually, first you have to start with an extraction. You can buy fancy extractors from WR or whatever. But also, you can go to Home Depot and buy a Dremel tool. That's a wonderful extraction tool. Actually, it has all kinds of attachments which allow you to grind bark, twigs, leaves. You can do it all in a scintillation vial. The battery will last you for 2 days. You can recharge it from, again, the cigarette lighter. And it's just a wonderful grinding tool. I strongly recommend it if you don't have large budgets in your lab. And so then, and it's just some simple assays. Like, for example, proteases and protease inhibitors. Again, for antivirals and cardiovascular protease inhibitors are also very important. A lot of skin type applications, also skin aging is connected to that. And again, you can develop a high throughput screen and transgenic whatever things, and this is expensive. Or you can basically go and get the thrown away X-ray film or photography film. That's becoming now a little more difficult because of the digital cameras, but still there's lots of photography film there. Why do you want photography film? Because all of it is covered with a gelatin. It's a protein. It's a wonderful source of just standardized protein. It's used to impregnate the silver grains. So now you have your plate, which has a very wonderful sort of layer of protein in it. You can try it at home. You can take papaya juice or pineapple juice, just spot it on X-ray film. It will burn a hole because proteases in those juices, will work. And now, if you want to, for example, find a trypsin inhibitor, chymotrypsin inhibitor, all you have to do is to now add it to your plant extract and see that there's no more hole. So you know now your extract has some kind of inhibitor. And that's very simple and it's very cost effective. The algal reagent, glucosidases, amylases, some wonderful, well-forgotten colorimetric tests for a lot of pharmacologically relevant activities out there. This is for glucosidases and amylases, important for diabetes. Beautiful colorimetric reaction, colorful reaction, almost instant, very cheap reagents, but gives you something important. Well, antimicrobial. Well, again, you can go to the sterile conditions and fermenters and autoclaves, which we all have, grow your cultures and then screen them. But somebody just figured out that basically the best inoculum we have for opportunistic pathogen is our saliva. We always carry a wonderful source of inoculum with us, which actually has a lot of relevant organisms in it. And just students have fun. They just spit in a test tube, plate this, and then they add plant extracts and they see what grows. It's easy, and that's always a fun part of Gimmicks training. It's just when everybody starts spitting in the test tubes. So anyhow, it's been a major, very underfunded activity, but which we carried whatever we had, research funds left, we would do that. We've done Gimmicks training. We actually trained people in 12 different countries. And usually countries, even poor countries, would pay half of that because we just didn't have enough resources. It's for us to stay there. We have trainers, we have graduate students who are very dedicated. All countries in red, we actually have conducted a week-long training sessions on Gimmicks technologies, which are now using Gimmicks assays and screens in their countries. Again, it all started with Central Asia, and we now have some Gimmicks technologies which have been transferred there. Uzbekistan, Kyrgyzstan, Kazakhstan, Tajikistan. In Africa, 2005, I think Tanzania was one of the first training workshops. Georgie Fear, which used to be a graduate student with us. Jeff Kellogg is here he's now in North Carolina State. It's been a lot of fun. 3 days, we teach courses, we teach vouchering, botanical vouchering, and those things which are essential. We determine what country needs. Then for 2 days, with the scientists, with students usually, we go to the field, we just take plants, we grind them with a Dremel tool, and we do all those assays. And then we leave them materials which are enough for 500 assays, and then we can even supply, but nothing there is expensive. This is actually 2 years ago, and this is Brittany Graff, who is in charge of training now. She's in my lab. She's a graduate student, has her own project, but she is always there when training needs to be done. This has been done in the University of Nairobi, and those are students from Kenya and Uganda, and faculty too. Actually, Ugandans, they endured 20-hour drive through the night just to be on this workshop. It was quite humbling for us. And now they have active programs both in Kenya and Uganda. This is the one which really, I think, one of the most active ones, and this is a Gimmicks program in Botswana. Each country has freedom how they want to use it. Some countries use it to validate, some countries use it to discover. Botswana uses Gimmicks and to really connect science with traditional knowledge. There's lots of traditional healers there in the bush in Botswana, which supposedly, been treating all kinds of diseases with herbal potions and stuff. And there's been a lot of mistrust between scientists in universities and these traditional healers. And a lot of people go to them. I mean, let's just face it. And they basically created, for the first time, this workshop that says, okay, traditional healers, come, we'll teach you how to actually show people that your plants, your extract, your potions may be active. We're going to test them. We're going to teach you how to test them. It's going to be simple. So they had this several major workshops with traditional healers. They came, and it was just such a, again, rewarding experience. And when they finished, I mean, this is Ngaka Themba Maun, was one of those traditional healers. He was just so happy. He loved it. So, you know, his statement, "Now I heal with pride." And there's this other couple who came from this tiny village said, "Well, the day you tasted our plants, I was very, very happy." And a lot of activities, we absolutely was validated, and particularly the antimicrobial, it's really, it was not random, I can tell you. We don't want to see the data unless the country wants us to see the data. We say we don't want to see the data because it's yours. We only see it if they want us to see it. So, Gimmicks Ecuador, I mean, that was a really wonderful program. Still is quite active. We've done training. We actually trained villagers from the village and university professors in the same team. And then they went out and collected in the rainforest. Manuel Baldion, this is here, dear friend and a colleague. Because of Gimmicks, he actually became the Minister of Science and Technology of Ecuador. He is no longer that person. But there we've organized, because of Manuel and his position, this wonderful biodiversity workshop 2 years ago in Quito, which truly brought some of the best scholars and scientists trying to. And one of the mission also was to protect the Tiputini region of Ecuador from oil drilling. You probably have heard they have the most biodiverse region in the world, and it's now in danger. Well, country needs money. I can't really blame them. And so we were able, and this is really a photograph I'm very proud of, because sort of two of my other role models. This is Ed Wilson from Harvard, and this is Peter Raven, probably the most famous living botanist. They wrote extensively on evolution, but never been to Galapagos before. So this is all of us. Look at this giant tortoise there. That was really a very, very successful forum, and we all went to Galapagos. It was just wonderful learning from those people who wrote so much about it. Gimmicks Bhutan. We were invited by the royal government. Somehow they heard about us. Bhutan is a very secretive society, still not quite open, but a remarkable tradition of traditional medicine, because they're between China and India, sort of squeezed in there. We have a wonderful training class there, and just love the country. They produce a lot of botanicals. They export them. They use some of the screens just to validate and standardize for antimicrobial activity, that activity, so forth. And this is probably one of the successes, again, which this is Gimmicks Mediterranean. This actually received money. This received 2 million euros 2 years ago, still active. None of that, unfortunately, can come to the US. Europeans don't like to share their resources, so we can just go there sort of as statesmen, as fathers. But this is still a remarkable partnership to use Gimmicks screens to look in the flora of Mediterranean region. It brought together Israelis, Palestinian Authority, Greece, and Spain. And we had our second workshop, and this is the first one in Jerusalem. Second one was in Spain about 6 months ago. A good friend and colleague of mine, Tola Friedlander here. Where is Tola? He's the president of Hadassah College, and really put this team together. And it is just remarkable to see how science can erase all kinds of political problems and political difficulties, and how well can people work together if they speak the language of science, particularly obvious because Israelis and Palestinians, for example. I mean, this just make a wonderful team. So, Gimmicks, we had 2 programs in the US as well. Gimmicks Alaska, again, this is traditional knowledge. This is working with Native Americans and children who don't want to listen to their grandparents who tell them about the value of traditional botanicals and shrubs and berries. And this is the way to validate it. We've taught kids those screens in high schools, and first, of course, they don't want to have anything to do with them, and then you start pipetting and colors appear, and then slowly move closer and closer, and then they're happy to do it. And we had that in North Dakota also with United Tribal College in North Dakota, and working with Native Americans. Again, trying to validate and maintain traditional knowledge. That's funded. That's actually been funded by USDA, EPA, and Marian Lyle actually is a PI in those. So this is it. I mean, one of the messages I'm bringing you, we take everybody. If you have some good ideas, you want to be a part of Gimmicks, we need you. It's a way to spread knowledge. If you know how to get money to fund some of that, because we really need new screens, and many countries really want this. I get letters all the time, but we just don't have, possibility sometimes to send trainers to everywhere where we need to go. Places like Chile, we've done the most recent training. Fine, I mean, they have money, they fly us in. But, you know, poor countries in Africa and South America just cannot do that. So anyhow, would love to have your university and students as part of Gimmicks. We're very flexible. And this is it. This is just a member of our team. I mentioned the work of many of them. I think too many to really be able to name. They're all wonderful people. They're really responsible for the data, which I showed you, and then they had to spend many hours explaining to me what it means so I can actually come here and try to impress you with that. Thank you very much.